But to keep us on time, because we also have a number of other presentations as well as our panel at the end, we're going to rock on to the good Dr. Charles Glabe, the professor of molecular biology and biochemistry at the School of Biological Sciences. Clearly an underachiever, not. Received his Ph.D. from the University of California, Davis. Postdoctoral training at the Johns Hopkins University School of Medicine, the UC San Francisco School of Medicine. Research program is focused on amyloid structure, its conformational polymorphisms, and the role of different types of amyloid in disease pathogenesis. Discovered that fibrils and prefibrillary oligomers represent alternative aggregation pathways from many different types of amyloids, and that they have distinct underlying structural motifs that are generic to the particular aggregation state. And he said I could actually think of all kinds of complementary things to say, but since we're in a hurry, I'm going to let you just imagine a couple of wonderful things, apply them to him, and listen to some really wonderful science. Doctor?
Well, thank you very much. Thank you for being here. So I'm going to talk today about the role of amyloid. So this is something you've heard about a lot today in Alzheimer's disease, about its role in the development of the disease and also as a target for the treatment of the disease. So for those of you who are wondering about whether you'd want to be in the research symposium or in the clinical, this is what I'm going to talk about. So I'm going to give you a brief history of AD that is selective and that of senile dementia. I'm also going to talk about the familial or inherited forms of AD and how this led to the development of the amyloid cascade hypothesis, which has formed the basis for most of the drugs that have been tested in human clinical trials over the last 10, 15 years. And I'm going to talk about therapeutic strategies that were based on this and their failure in human clinical trials, but I'll also tell you about one that is the first human clinical trial that has led to a disease-modifying therapy in Alzheimer's disease was announced by Eli Lilly. And unfortunately, for the patient, it actually made Alzheimer's disease worse, but it did modify the disease, and I think that's been very insightful for basic research because it's telling us that we're thinking about the disease mechanisms backwards. So what I'm going to do is I'll tell you, based on this, there's an alternative way you can order all of these facts that have led to the development of the amyloid cascade hypothesis into an alternative amyloid hypothesis, and it's based on the intraneural accumulation of amyloid prior to neuronal death, which leads to the formation of a neuritic plaque. So this alternative model proposes that there's a one-to-one relationship between a dying neuron and a neuritic plaque. And then at last, I'll talk about new potential therapeutic strategies based on a different view of the mechanisms of amyloid pathogenesis in Alzheimer's disease. So as you all know, this is August D., who was Alzheimer's first patient in 1905. And this is what basically they saw, and you've seen this exact same picture multiple times today, that in Alzheimer's brain there are amyloid beta deposits known as senile plaques that are shown in this brown stain. And there's also this tau that's shown in black that looks like these flame-shaped or arrowhead-like figures. And 20 years ago, those of you who've been following this work for a long period of time know that basic researchers argued for many years about which one of these was more important. Was the amyloid versus the tau? And now we know that this tau is just forming another different flavor of amyloid. So these are both amyloids, and when they fit the biochemical definition of a amyloid, which is a misfolded protein that aggregates into a beta sheet that can extend infinitely into very long structures by addition of another protein onto the end of it by forming intermolecular hydrogen bonds with the underlying peptide chains below it. So both of these are amyloid, so there's no argument about what they are, but there's still plenty of controversy about how they're bad, what makes them bad, and why they're associated with disease. But the presence of amyloid in diseases is very fundamental because not only do we find two different kinds of amyloid in Alzheimer's disease, it's very common that in Alzheimer's disease there'll be yet a third type, which would be alpha-synuclein amyloid, which is commonly associated with Parkinson's disease, but it occurs in about 30% of Alzheimer's patients, or maybe a little more, in the Lewy body variant of the disease. So we may have multiple different types of amyloid in Alzheimer's disease, where in other diseases they might be a pure flavor of only one type of amyloid. So here's a little brief history. It's the timeline of the development of milestones in Alzheimer's disease starting in 1907 when Alzheimer's was originally described by Dr. Alzheimer. And he described plaques and tangles in a middle-aged woman that had dementia. And what was unusual about her is she was relatively young. I'm not sure how old she was, but she was certainly not more than, I think she was in her mid-50s or something. So the concept of Alzheimer's disease originally was associated with people that were relatively young that had dementia. Now, for centuries, it's been known that as you got older, it was very common that you would have senile dementia. But it was between the time of the discovery of Alzheimer's disease in 1907 until the next period in timeline, 60 years later, Bob Terry and Henry Wisniewski discovered that the pathology of Alzheimer's disease is identical to that of senile dementia. So now this problem went from having a few thousand people that had the disease to being a problem of millions of people, and that caught the attention of drug companies because now this is a very significant disease and it's going to affect a very large percent of the population, and it gives us opportunities to develop drugs that are going to make a lot of money. So between this time, 1967, the next milestone is 1976. Peter Davies reported the loss of cholinergic function in the Alzheimer brain, and it initiated the development of a major class of drugs that are currently approved for Alzheimer's disease, including Aricept and Exelon. So this was based on a discovery in 1976, and it wasn't until maybe, almost 20 years later that these drugs came on the market, and they are the drugs that are currently prescribed. And those of you who know about these drugs, they're not wonderfully effective, and one of the things I like about them particularly is that you can think of these drugs as funding the next generation of research in the pharmaceutical industry. So hopefully we'll get some better ones. And that's a kind of a charitable interpretation of that. Now, the next milestone in the timeline of Alzheimer's disease pathology was the discovery by George Glenner where he isolated the A-beta peptide, which is the major component of plaques. But interestingly, he never isolated this from Alzheimer's disease brain plaques. He isolated it from the cerebrovasculature, and he made a guess that this was similar because it had the same kind of staining or tinctorial properties that the plaques did. So nobody could ever isolate the A-beta peptide from Alzheimer's disease plaques, but they could show that the A-beta peptide that they isolated from the vascular amyloid deposits was immunologically the same as what was being deposited in the plaques in Alzheimer's disease. So this started the molecular biology era because now we knew the identity of the molecules that were accumulating in the lesion. Now, soon after the identification of the A-beta peptide as the major component of plaque, then it was discovered that the neurofibrillary tangles contain the protein tau a year later, and then in '87, it was found that this A-beta peptide that accumulates in plaques is derived from a much larger precursor protein that is embedded in the membrane that surrounds the cell. So this was a lot of work, 85, 86, 87. The research in the molecular pathology of Alzheimer's disease is becoming exponential at this point. So soon after that, Alison Goad and John Hardy discovered mutations that occur within the amyloid precursor protein, or APP, that are associated with the inherited forms of Alzheimer's disease, which was the original Alzheimer's disease that was described by Dr. Alzheimer. So they showed that mutations in the APP gene could give rise to forms where the Alzheimer's was inherited in a dominant fashion. Whereas if you inherited a bad allele or bad copy of the gene, you would get the disease, 100% penetrant. And similarly, then after there was discovered that mutations in the APP gene were associated with Alzheimer's disease, this implied that the A-beta peptide is on the causal side of the disease. It causes the disease rather than being an effect of the disease, okay? So soon after that, Peter St. George Hislop discovered mutations in a separate gene, which is known as presenilin. And this is associated with the vast majority of inherited forms of the disease. So it's mutations in presenilin are the mutations that are most commonly found in inherited forms of the disease. And subsequent work then showed that these mutations work because presenilin is what proteolytically processes this larger APP to the A beta peptide. So we've got two different things that tell you that the amyloid is on the causal side of the disease. Because if you get a mutation in either the protein that's the precursor to A beta or the enzyme that makes A beta out of APP, you are going to get the disease early in your life 100% of the time that you have the bad mutation. Okay, so this formed the basis for the amyloid cascade hypothesis because it was such a powerful indication that the A beta peptide is causing the disease rather than being an effect of the disease. And based on this, then, the pharmaceutical companies have developed drugs that are basically all designed to prevent the production of this A beta peptide. And here we had the spectacular failure of the Eli Lilly semaglutide, but this was the first drug that has ever been shown in human clinical trials to modify the course of the human Alzheimer's disease. Unfortunately for the patient, it actually made the patient significantly cognitively worse than those people that took the sugar pill or the placebo. But this told us something very important. And then recently we had the failure of a monoclonal antibody, bapineuzumab, and just last month there was a failure of another class of drug called a BACE inhibitor because it was withdrawn because the toxic side effects were too severe to continue the clinical trial. So this is basically the timeline of our understanding of the molecular pathogenesis of the disease and the basis for the development of drugs to try to treat this disease in humans. Now, a little more detail about how you get A beta produced from APP. It's derived by cutting this much larger precursor protein in two places. So this is a cartoon or schematic diagram of the APP molecule. This here is the plasma membrane of the cell in which this protein is embedded and is floating around and freely diffusing in the membrane. There's an N end and a C end, and we'll talk about this a little more. So this is the amino terminus of protein. This is the carboxy terminus. All proteins have these two different ends, which means that they have a direction to them. There's a polarity to the protein. And the A beta peptide is this small 42 or 40 residue piece of a much larger protein that can be anywhere from 695 amino acids to 750. So it's cutting out a very small piece of this much larger precursor protein. Now below, this red region is expanded here. You can see the entire sequence, and these letters correspond to the individual amino acids that make up the A beta peptide. And these arrows are different places where the enzymes cut this, and you have to cut the protein in two places. You have to cut it on the amino terminal side at this site with the Greek letter beta, and you have to cut it on the other side with this Greek letter gamma to release this A beta peptide to make soluble A beta. So it turns out that these sites, the gamma 40, gamma 42, and epsilon, are the sites at which this presenilin, which is the enzyme that actually cuts this. Okay, so these are the places in which mutations occur that affect the proteolytic processing to make more of this protein that accumulates in the disease. So based on the genetics then, it led to the development of the amyloid cascade hypothesis because the genes that are associated with familial Alzheimer's disease either are the precursor for A beta itself or the two different isoforms of the enzyme that proteolytically cleave this to make A beta. And that includes presenilin 1, which is also known as gamma secretase, and there's a slightly different form called presenilin 2 that is also gamma secretase that cuts this protein. Now, this shows the development of the amyloid cascade hypothesis, and you don't really need to read this in order to understand it, but in the earlier forms of this cascade hypothesis in 2002 and 2011 hasn't changed that much. And you can tell how clearly scientists understand the pathways leading to pathology because it's inversely proportional to the number of arrows that are in their model, okay? So if you look at this model, there's 10 arrows here, which means that they only understand about 10% of what's really going on here, okay? Almost 10 years later, we're still got about 10 arrows, but at least we're being honest now. There's some question marks. Here we were confident, but now we know we really don't know about some of the important questions like what aggregate stress means. And the other things you can see is some of these things are very vague in terms of what these, the definitions of these things. At the bottom, it's always dementia, okay? So what this means is we really don't understand the ways in which the amyloid A beta peptide is actually causing pathology. Now, it hasn't kept scientists from proposing things, but the only way you really know if it's really true or not is if you make a drug that targets a particular step in this pathway and it goes into human clinical trial and it actually cures the disease. And as you know, that hasn't happened yet. But that doesn't mean that we haven't learned anything about what's causing the disease. Now, this shows you where the mutations lie within the amyloid precursor protein. So there's basically 3 different classes of mutations. There's mutations that occur at this beta cleavage site. So see these clever little scissors. This is the enzyme that cuts on the amino terminal side. And these mutations, what they do is they give rise to increases in the total amount of A beta that's made by this beta secretase in cleavage. So it increases the amount of beta secretase cleavage, and it just increases the total amount of A beta made about 5 to 8-fold. Now, mutations in the middle are very different. These mutations don't necessarily affect the proteolytic cutting of this at all, but what they do is the amino acid substitutions increase the rate at which the peptide aggregates and oligomerizes and sticks to itself. So this is fundamental. All of these amyloids stick together to form things that are highly insoluble, and that's the key distinction between a normal protein and a pathological one. And it's these mutations enhance the ability to aggregate. And then lastly, the ones that occur within the membrane, transmembrane region of the protein act by a different mechanism. They influence the places at which the gamma secretase will cut. So gamma secretase starts cutting over here, and it starts chopping away at this end and taking progressively more bites from this end and making this A beta peptide shorter. So the better that gamma secretase works, the shorter it gets. And the mutations make gamma secretase work less efficiently. So it can only make 1 bite or maybe 2 bites, and what it ends up doing is it makes A beta peptides that are longer. So it favors the production of A beta 42 and actually even longer peptides over A beta 40 and shorter peptides. And the longer peptides aggregate much faster, again, pointing to the key feature that the thing that distinguishes the disease from the normal state is the aggregation, okay? So why is aggregation important? And we'll come back to that in a minute. Now, when these presenilins were first discovered, it was found that all of these presenilin mutations increase the longer form and compared to the amount of the shorter form. And since then that the longer form aggregates much faster, this suggested then that the production of the longer form is really the culprit in Alzheimer's disease. And this formed the basis for many of the therapies that have gone to human clinical trial based on the idea that we wanted to suppress the production of this soluble A beta 42. So here's basically all of the drugs that have been tried or developed and have gone to human clinical trial based on this traditional amyloid cascade hypothesis. So they include drugs that prevent, that enhance or activate this alpha secretase cleavage. So this cleavage cuts the A beta peptide right in the middle. I'll show you where. Just to remind you, it cuts here. So if it cuts right in the middle of the A beta peptide, it prevents the A beta peptide from being generated. So alpha cleavage would be good. So this would be a good cleavage, this would be bad, and this would be bad, according to the amyloid cascade hypothesis. So the drugs that have been designed either enhance this cleavage, or inhibit this bad cleavage, or inhibit this cleavage that was believed to be bad. Okay? So we'll go back. So the other classes of disease are beta secretase inhibitors. They inhibit the cleavage by base, and so this was just the one that was recently tried, and it was halted because it had so many side effects and toxicities that the trial had to be halted. The other ones are gamma secretase inhibitors, and this were several here. This LY compound, and I apologize for it being so small, is the semagasistat that was tried in 2010, and we'll come back to that in a little bit. And then there's another class of drugs that I want to call your attention to, and those are called the gamma secretase modulators. So rather than preventing gamma secretase cleavage from occurring, they want to make the gamma secretase more active and to make it take many more bites off of the tail of that A beta peptide so that it becomes shorter and shorter. So instead of making a peptide that aggregates fast, that ends at residue 42, it makes shorter peptides of 40 and 38 and 39 that are much less likely to aggregate. So there's classes of drugs that are coming down the pipeway called gamma secretase modulators that will do these, and I don't think we have any data yet back from clinical trials of gamma secretase modulators, but you'll see in a minute, I think these have a better chance of winning and actually succeeding than the gamma secretase inhibitors. And then, after the A beta is actually produced, there's a whole set of classes of drugs that are designed to prevent the aggregation. So these are aggregation inhibitors. There's also classes of drugs like the A beta immunotherapy, either active immunotherapy or passive immunotherapy, that are designed to produce antibodies that bind to these aggregates and cause them to be taken up by microglia and to be removed from the brain. So all of these things have gone to trial. So this includes bapineuzumab, which is the Eli Lilly antibody that failed in the clinic last year. And it includes solanezumab, which also failed. Bapineuzumab was from Johnson & Johnson, and solanezumab was Eli Lilly compound. So all of these things have failed, except for the semagasistat that was actually a spectacular because it was the first disease-modifying therapy, but the bad news was because it failed to, it's bad for the patient because it made the disease worse. So let's look at the record of the drugs that were based on the amyloid cascade hypothesis in the last 13 years. So we've had, I just looked, and the number so far is now 110. So this was based on September 14th. Almost a year ago, it was 101. But if you look, then 101 have gone to human clinical trials, have been tested on humans, and none of them have worked. The 3 Alzheimer's drugs that have been approved were based on the cholinergic hypothesis, and as you know, they're not very wonderful drugs. So we have a loss ratio of one drug that actually is approved for human use, that even including the ones that don't work very well, it's a 1 to 34. So we haven't had any winners yet. Now this is the one that's interesting because this drug actually is the first disease-modifying therapy. When it was tried in a very large phase 3 clinical trials, it actually led to a significant worsening of clinical measures of cognition and the ability to perform activities of daily living. Okay? So these are primary pre-mortem clinical measures of the progression of Alzheimer's disease. Now, the pharmaceutical people and a lot of researchers, we were doing a lot of hand-waving about what this result means. So some people are claiming these are side effects. But these aren't side effects. If you're taking an anti-cancer drug and your hair falls out, your hair falling out is a side effect, right? Unrelated to its effect on the tumor drug. If you're taking an immunosuppressive drug because you have a kidney transplant and you get a fungal infection, that's a side effect. But when you actually get a worsening of the disease that you're trying to improve, that's not a side effect. That's the primary effect. So it's telling you it's modifying the disease and it's doing it in the opposite way of what you intended. And I think that's very significant for scientists because the simplest interpretation of that, and you know, as scientists, we like Occam's razor. Occam's razor says we should accept the simplest interpretation. The simplest interpretation of that is we're thinking about the disease mechanism backwards. So rather than inhibiting gamma secretase, we should be promoting it. So rather than secreted A beta being bad, it's good because it predicts then that it's the precursor to that, which is the the piece of A beta that's been cleaved by beta secretase that has not been cleaved yet by gamma secretase is really the bad guy, okay? So that's the alternative view, and I want to convince you that there's some data that this may be true because it's important, because if you understand the mechanism of the disease, then you can develop drugs that actually work, and this is where I think the field is headed, except for they don't. Very many people are reluctant to give up the old way of thinking about it because they've been working on this for 20 years. And as scientists, we like to take the baggage of our previous success with us to our next experiment. So we've got to give up that and think about new alternative mechanism by which we can reorder the same facts about the causality of amyloid into a more legitimate and honest and truthful mechanism of pathogenesis. So again, the big question right now in the field for people who are trying to develop drugs that are effective is, why is this amyloid cascade hypothesis failed so miserably in the clinic? So one is that the amyloid cascade is wrong and the A beta is not important. And a lot of people really felt this way. So A beta is not important, but it ignores all the data from the human genetics. The other thing is simpler. We're just not thinking about the amyloid hypothesis correctly. In fact, we're thinking about it backwards. So there is another way of ordering all these into a model or a hypothesis that can account for the primacy of amyloid in causing the disease, being on the causal side, and yet accounting for the spectacular failure of semaglutide and the other drugs that are based on lowering soluble secreted A beta. And that basically is this recent discovery that all of these amyloid-related diseases, whether we're talking about Alzheimer's disease, ALS, Parkinson's disease, Huntington's disease, behave like prions. Now, those of you who've heard about prions know that prions have this peculiar property of being able to infect cells. So examples of prions are mad cow's disease, Creutzfeldt-Jakob disease, and other diseases that were like Kuru that were transmitted by actually eating infected meat from people that had prions, or in the case of mad cow's disease, animal meat that had been infected with these prions. But it turns out that all of these diseases have a prion-like property in that the disease can be transmitted laterally throughout the brain by the cell-to-cell transfer of amyloid aggregates or oligomers that are taken up by the next cell or the neighboring cell and cause the aggregation process in an otherwise normal cell. So that would include Alzheimer's disease, tauopathies, Parkinson's disease, light chain amyloidosis, a whole bunch of diseases that are all similar to Alzheimer's disease in that they are age-related diseases that are neurodegenerative. Now, the way that the prion model works is that you take a normal protein, a normally folded protein which has a normal job, and in this case of PRP, which is the precursor, prion precursor, that's what PRP stands for, it's normally alpha helical. So helical means happy. And when it misfolds, it adopts a beta sheet structure which allows it to aggregate with other misfolded proteins of the same type to form these beta sheet intermolecular stuck aggregates. And the important thing about prions, that is true about all of them, is it's protease resistant. So in this model, notice we have one arrow in the diagram. We go from normal to aggregate and protease resistance. So the simpler your diagram, the more you know about the mechanism, right? It's inversely proportional. This is a one-arrow model. So this is the way it works in a little more detail. We have the native structure. The native structure misfolds and makes it look like a pyramid. The consequence of this misfolding is now it can stick to itself in an infinite fashion. It can grow and grow and grow by accretion of more of these misfolded, and this could go on and on and on. And it turns out that it goes to a certain stage where it becomes infectious, and what happens is this infectious form can replicate. So it grows to a certain size, and then as it gets too big, it accidentally fractures or breaks into two pieces, Now we have two pieces after this breaks apart, and each of these can grow, and they grow to a certain size, and then they fracture, so they replicate by this binary fracturing fission mechanism, and this is how they get disseminated widely through the brain, and this is why they accumulate. And they accumulate simply due to the fact that they're protease resistant. So does this go on in A beta and APP? And the answer is yes, it does. So it's seeding dependent. So what we do in this simple experiment is to take A beta 42 oligomers and we add them to cells that are expressing the amyloid precursor protein. And we look to see what happens to the amyloid precursor protein. And what happens is that these are the cells that are expressing APP. These are cells that don't express APP. And you can see that when we treat them with A beta 42 aggregates, all of this amyloid precursor protein is now become aggregated and insoluble. And this arrow points to the beta secretase cleavage fragment of APP, which is the immediate substrate for gamma secretase. This is the bad guy, and this is now aggregated. Now, the other fundamental thing about prions is once it's aggregated, the cell has no way of degrading it because it can only degrade linear protein molecules that are spaghetti-like, because the proteases have to be able to be able to grab onto it to be able to cut it. And what we showed further is that this material that accumulates is actually resistant to proteolysis. So you can see we've purified this material that was made by the cells, and at time 0 it's fairly high molecular weight. It has a very broad spectrum of sizes, and the closer you get to the top, the larger it is. And as you digest it with this very broad spectrum protease, you can see that the size drops down to that of 4 kilodalton A beta, but it doesn't go away, okay? So what's going on in the disease then is a very simple molecule. Again, this is my model, which is based on the prion model of Stan Prusner. You've got one arrow, it's the aggregation of A beta 42, and that leads to protease resistant. And it doesn't matter how much of it's protease resistant. It could be 50%, it could be 1%, or 0.1%, but if there's some fraction that the cell can't get rid of, it's going to accumulate over time, and then at some point, the cell will fill up with this amyloid and won't be able to live. So the way this then works is once this is nucleated, then the APP fragments that would be normally degraded will add on to the end of these aggregates and extend them by one polypeptide. And then the cell will digest away the parts of the amyloid precursor protein that it can, and it will leave behind the APP molecule. And then this leads to the growth of aggregates. So what I want to get to is there any evidence that that actually goes on in the human brain? Okay? And it's based on the fact that antibodies can detect these pathologically misfolded states because they are specific for aggregation, and they don't recognize the native protein or the monomeric protein. They only recognize the protein after it's aggregated and become insoluble. So here's an antibody that we've made that specifically recognizes an aggregation-specific fibril epitope. So you can see here that this antibody does not recognize A beta 42, alpha-synuclein, or islet amyloid in its monomeric state at time 0, but as fibrils evolve in the test tube, then you can see that the antibody reacts with these protein samples as it aggregates. This Western blot also shows that it only recognizes aggregates that are about the size of dimer. Monomer would run down here, so it's either dimer, trimer, tetramer, and things that are really big that are recognized by this particular antibody. So what's interesting about this is this antibody shows that the first time that the APP molecule misfolds is inside the neuron, well before there's any other pathology and well before there's any plaque deposition. So at 3 months in this triple transgene mouse that was developed by Frank Laferla, and you heard about it from Matt Blurton, you can see that there's an elevation of the expression of APP, shown in green, in the neurons, but this misfolding or aggregation-specific antibody is in red. And you can see that the red only accumulates at 10 months rather than 3 months. So the red is now co-localized with the APP and the A beta molecules around the nucleus, and the nucleus is stained in blue here. So the blue dye actually corresponds to the nuclear, the nuclei of the neurons here. So you can see that this shows that the first time that the protein aggregates is when it is inside the neuron and it has not been completely metabolized to A beta yet, because some of these molecules are still APP and carboxy terminal fragments of APP. And then 2 months later, most of this is accumulating in the nucleus. So you can see that the red is separated from the green, and now it is found inside the nucleus. And then 2 months later, all this nuclear immunoreactivity is gone, and now we see the first extracellular plaques. So the timing in the transgenic animal shows it misfolds first inside the neuron, it accumulates inside the nucleus of the neuron, and later it appears as the extracellular plaque. Okay? So do you see the same? When the question is, what happens between this time when it's inside the neuron and this time where it's outside the neuron? How does it get outside of the neuron into the extracellular space? And the way it does it is the neuron dies and degenerates and forms something called a neuritic plaque. So this shows the DNA staining. So these are neuritic plaques shown by this arrow. And you see the exact same spatial relationship of the nuclear amyloid staining in red, which is co-localized with the DNA, this diffuse DNA staining, and it's surrounded by the amyloid precursor protein carboxy terminus. So this shows that those neurons that had that nuclear amyloid and were surrounded by APP and misfolded APP then have the same kind of spatial orientation in the dystrophic neurites around the plaque. So this shows that the neurites are that the neuritic plaques are derived from single neurons that had accumulated this misfolded APP molecules. And this is just further evidence that they're neuronal, because there's a neuronal nuclear marker in red here. So these are normal neuronal nuclei, and the degenerating ones are shown here. You can see that each of these plaques, these neuritic plaques, has the nuclear marker nu-N in the center of them as well. So it has the DNA, the misfolded amyloid, and the nu-N nuclear marker in the center of these neuritic plaques, meaning that these are dying neurons that had originally accumulated this intracellular amyloid. So the question is, does it happen in humans? And the answer is yes, it does. You see the exact same kind of thing, the misfolded APP accumulating in the nucleus, and it's surrounded by these misfolded APP molecules in the dystrophic neurites, and there's DNA in the center of the human neuritic plaques as well. And the interesting thing about this is that this nuclear pathology and intracellular amyloid accumulation occurs very early in Alzheimer's disease and occurs prior to dementia in human Alzheimer's disease, and there's an increasing awareness that the disease occurs much earlier than when you first see dementia. And this is what we see with this nuclear pathology. Most of these patients that have this are non-demented at this point. And the other thing is it goes down as plaques evolve, then this intraneuronal pathology decreases, as you would expect, because they become, the infected neurons become plaques. So this is an alternative hypothesis based on interneuronal accumulation of the APP in the nucleus, followed by the lysis and dying of the neuron to form the neuritic plaque. So in summary then, the conformation-dependent fibril-specific monoclonal antibody M78 stains nuclei in aged human brain and intracellular amyloid, and is correlated with intracellular APP and A beta immunoreactivity. And it co-localizes with APP-CTF immunoreactivity, suggesting that this is the real culprit that we should be targeting in the disease. And we observe the same spatial relationship in neuritic plaques and senile plaques, indicating that senile plaques are initiated by the death or demise of single neurons that have accumulated this misfolded APP. So this has implications for the development of drugs. First of all, it can explain why the semagacistat clinical trial made things worse. By inhibiting the disposal of this APP-CTF, you made it more likely to aggregate and accumulate inside neurons. So this is a prediction. If we looked in the brains of these patients that were treated with semagacistat, we'd see a massive increase in the amount of intracellular or interneuronal APP staining. But it also has some therapeutic implications as well. Because of the fact that this intracellular amyloid accumulation becomes early, much before significant cognitive impairment, it suggests that we must start treatment early on in order if we're going to arrest the disease progression. So we might have to start treatment 10 years prior to any signs of cognitive impairment. So we're going to need therapies that are very safe. Now, the important thing is that the same kind of strategies that have been tested in mild to moderate might actually work if they're started earlier, so they can prevent this kind of pathology from initiating and being transmitted in the between cell to cell. And then inhibitors of the aggregation process would need to gain access inside the cell because it looks like the pathology is going on by aggregating inside the cell rather than outside the cell. And lastly, extracellular aggregation inhibitors and immunotherapy may work by preventing the cell-to-cell spread by this seeding mechanism. And then lastly, not shown here, is I think that the gamma secretase modulators that improve the activity of gamma secretase and actually dispose of the CTF much more efficiently, giving rise to shorter one, are the ones that I would bet on that are going to win in the clinic. Okay? And lastly, I'd like to thank the people that actually did the work. And I'd like to thank the people that helped fund this, including these private foundations that have been helpful in giving us early stage work on new models and new types of therapies for disease. Thank you very much for your attention. Any questions? Yeah.
I tried all kinds of stuff and I've got nowhere.
Yeah.
And so finally I threw everything away, and I feel better the last 6 months than I've ever had the rest of my life. And I'm not taking anything. So
I'm not
sometimes
surprised.
I beg your pardon?
I'm not surprised.
Okay.
I try to under-medicate myself. I think it's a good strategy. Unless it's recreational, and then all bets are off. Yeah.
So would LMTX fall, in terms of what's in clinical trials now, especially phase 3 trials, would LMTX be what you would bet on, or what, could you be more specific about what you would bet on?
I hate to admit my ignorance, but I don't know what LMTX is. Oh, the tau inhibitors. Well, I think tau is a different kind of amyloid.
Okay.
I think it's another amyloid. I think it's important that we're going to have to deal with tau too, but this, I only was considering a beta amyloid. But I think the same kind of things happen to tau, but it's in tau, you don't need to have any proteolytic processing to facilitate aggregation. It seems to aggregate fairly well on its own without any. I would bet on the gamma secretase modulators. Those are the ones. Oh, I don't know the names of these things. There's a whole bunch of them. So, but I don't know the trade names. I just know the generic class. So they're called GSMs or gamma secretase modulators.
Hi.
Yeah.
I'm back here. This is probably a silly question, although there probably aren't any silly questions. But you were talking a lot about immunotherapies, and as far I'm like not very well educated medically, but I'm on a biologic medicine to suppress my immune system. So
right.
Are the immunotherapies that you're talking about completely different than something like a biologic that works on the immune system systemically?
Well, first of all, there's no stupid question. The only stupid question is one that ends up on the test that you didn't ask. Okay. As somebody who teaches undergraduates. But immunotherapies are all biological because the antibodies are produced in living cells. The one you're taking for immunosuppression is very different than these types of antibodies. They just happen to be antibodies, but they have different targets. So the one you're taking, you know, I forget what its target is, but it's one of these inflammatory modulators that it neutralizes the activity. These other antibodies bind to the amyloid aggregates that are outside the cell, and they cause them to be either inactivated so they can't be taken up by another cell, or they cause them to be taken up by microglia and to be degraded if they can. So those are mainstream therapies that are able to clear plaques in humans, but they haven't shown any cognitive benefit. And my argument is that the reason they haven't worked is they're being worked too late. So instead of being added at a time where the disease is actually happening, they're being added at a time where after the bomb has gone off. So they're cleaning up the mess, but they're not helping the function.
Thank you very
Okay.
much.
Okay, thank you very much.